Thank you very much. I should give her a name. All right, we should like have a.
Amanda.
I can go for that. I like that.
I don't know.
Yeah.
Just pulled it out of the air.
I'm good with that. Emily. Bex was going for Emily. Emily's good too. How about a compound name? Emily Amanda. All right. Amila man. That doesn't have quite the same effect. Amanda. I like that. Okay. This look about right. Okay, so we should have a pretty fighting chance here of getting 9 finished, and then 11 will go quickly because there's a lot of recapitulation actually in 11 that we've already covered in 9. We just kind of expand it to neurons. So energy sources for muscle contraction. Well, some of this is kind of a yada thing, right? Because we kind of know where the energy comes from. We've been sort of talking about it a little bit the entire time. I mean, we saw how you energize the cross bridge formation and release and power stroke. It's all ATP driven. And so that's obviously part of it, right? So the majority of it is honestly ATP driven, right? Because of that requirement for the myosin heads and the detachment and the power stroke. So it's all basically tied into ATP and also into the hydrolysis of ATP, right? So as you kind of transform into ADP and then you kind of let the phosphate and then those sorts of things. It's also, however, required for the sodium potassium pump, right? Because you have to build your sodium concentration gradient and your potassium gradients in the first place in order to actually get a polarized membrane. That requires some ATP. Also, you need ATP for your active transport calcium pumps, right? In order to get your calcium pumped back into the sarcoplasmic reticulum. And so ATP is the primary energy source, which is, you know, the reason why, like when you're really active and you got a lot of muscle use, you oftentimes, like when you're working out or you're athletic or you're kind of training, you tend to have an appetite that goes along with it because you're requiring more energy because you're using it more for musculature and things of that nature. Now, generally speaking, most of the ATP is sort of transported. Some of it is made on site, but your skeletal muscles will kind of have a little bit of reservoir of ATP, not a lot. Basically, you store about as much ATP as you can use for about 5-6 seconds of contraction. So that's about it. So what does that mean? So that means if you plan on contracting for longer than 5 or 6 seconds, you better have another plan, right? So that's kind of where you get into the whole oxygenation and the metabolism thing. So basically, when you're talking about the majority of the ATP that you are consuming during physical exercise, most of it is actually being made at the time. So it's basically like made to order. Yes, right. So as your muscles are consuming the ATP, your mitochondria are busy belting it out, which is the reason why you require more oxygen when you're exercising, is because the oxygen is part of the cell respiration piece. It's making the ATP, which is part of what's happening in the mitochondria. So essentially, except for all of about 5 or 6 seconds worth of it, most of that ATP is being made on demand. Okay. So, so when you take a look at ATP, how do you make it? So there's a couple of different ways you can make it. Now, obviously, the big one that you're going to see that we think about mostly is aerobic respiration, right? So that's basically where you're breathing oxygen. And then you're going to be taking a molecule of glucose and you're going to be breaking that ultimately down into ATP. Of course, you have your waste products, your carbon dioxide, and your water, which are waste products. That's the reason why you exhale CO2 and water vapor, is because those are metabolic byproducts. So that one we know about. This is your most efficient version, right? So this is your gold standard. This is the reason right here why a lot of times doctors who are kind of interested in getting you in some sort of a physiological exercise program where you're doing yourself some good, oftentimes they'll say something like this. They'll say, when you're working out, make sure you work at your strenuous level is such that you can hold a conversation with somebody, right? So like if you're jogging, jog at a level where you can hold a conversation with somebody. Now, the doctors don't care whether or not you talk to somebody or if you're talking to yourself. right? They don't care about the conversation per se. The reason why they say that, like hold a conversation with somebody, is because if you're holding a conversation with somebody, that means you're breathing. That means you're getting enough oxygen, which means that you're at your maximum efficiency for aerobic respiration. So this is basically your engine when it's really running really, really well. And that has nothing but good associated with it, right? That's where your physiological magic fairy dust comes from, is when you're in that kind of sweet spot of your aerobic respiration. That also happens to be the level that usually you can maintain for a good long period of time, because it's low wear and tear, but it's high physiological value. And so your body can easily maintain this level usually for a pretty good long period of time without a lot of taxation on the actual body itself. It also happens to be typically right around where you start to get maximum benefit, not just in physiological metrics, but also things that like fat burning, energy consumption. So all of that is at your maximum efficiency. So like if you're trying to burn fat, then this is where you want to be. You want to be in your aerobic zone, right? You want to be breathing, right? And so obviously then within that context, there's going to be different levels, right, of that. So for instance, somebody who is not accustomed to aerobic exercise, but starts to exercise and maybe they're carrying a bit of weight, for them, carrying a conversation, breathing is going to be at a much lower activity level, right? Because there's a bit more taxation in terms of their physical conditioning, right? Because they're not as well conditioned. As say somebody who's active and who's doing it a long time, they can kind of run at a bit of a higher motor, so to speak, and still be able to kind of maintain a good conversation. So you have a lot of plasticity there, right? And so that's kind of what we mean about getting into shape, right? The idea about getting into shape is once you're into shape, is you can easily get into this particular zone itself and still be able to maintain this kind of high efficiency rate, right? So when you're out of shape, it's like, well, the level, your activity level to get into this place is like really, really low. So you're not quite there yet. You haven't trained your body to sort of exist at this level. And so it's just like anything else with your body, right? The more you kind of treat your body that this is the norm, then your body calibrates and adapts itself to that new norm. And then you kind of exist at that level. And that's what the doctors are wanting you to get to. This is the reason why the doctors say, I would rather you do something very low impact, but consistently, right? So whatever that is, just do it every day. If that means you're just doing 10-minute walk around the block, then do it every day. I don't care what it is, but just do it consistently, because of what I want your body to go from 0 to something above 0. And then we'll work from there, right? So once you get used to that above 0, then we can move it maybe a little higher than that, and then kind of continue that benefit. That's what they're going for, right? So that's kind of that sense of conditioning and things like that. However, what if you don't have oxygen? Okay, eventually, yes, you will die. Thank you, William. It's like, was this an easy test question? I think this was, right? I hope this was a 50-pointer, right? It's like, okay, so what happens if you're still breathing? Okay, so you're not dead yet, but your oxygen levels are not quite where your activity levels are. Correct, right? So right now is a good example. You're sitting, so your energy demands are very low right now because you're sitting and you're at rest. But if I tell you to jump up and start running as fast as you can up and down the hallway, all of a sudden your energy demand has gone way high in a very short period of time. But guess what? Your energy supply has not, right? And so this is the reason why in that short period of time, we have to have an alternative strategy, right? Because our respiratory system has to basically catch up to the rest of our system. Our muscles can jump out of the chair and run in a second, but our respiratory system is a little slow to follow, right? So the question is, if we're not breathing enough oxygen, so if the oxygen you're breathing as you're running up and down the hallway is calibrated for you sitting down at the chair, in the chair at rest, then how is it that you're able to generate enough energy to be able to run up and down the hallway? Because you're not clearly breathing enough oxygen, right? Well, the answer is it's the rest of these three, right? And the rest of these three basically are short-term anaerobic strategies. So this is like the sprinter situation, right? So when you don't have enough oxygen to support your energy demand, you still have to get up and run, right? Why is this a good idea? Do we really care about running up and down the hallway? No. Yeah, right. But if I let the lion in the room, then you'd better jump up and run, right? And not sit here and wait for your oxygen level. Oh, wait a minute, I need to run right now. Hold on. Okay, now I'm ready. Let's go, right? So that's not going to work. The lion's not going to sit there and be like, are you ready yet? Are we ready yet? Right? You're going to be chow, right? You're going to be lion chow. Okay. So obviously you got to get up and run fast. You got to do it now. And so this is your short-term coverage, and you have several different strategies to this. So for instance, the first one, you can convert ADP to ATP. And so these are basically just variations, right? So ATP is adenosine triphosphate, ADP is adenosine diphosphate, and then AMP is adenosine monophosphate. So it just depends on how many phosphates you've got on there. And so you can convert ADP to ATP with an enzyme, adenylate cyclase. Now, this is basically low output. So this is not like a long-term strategy, which is the reason why we're talking about just short-term coverage. But that's fine, right? Because you don't need a long-term strategy, because the idea is by the time you get to the end of the 40-meter dash, your oxygen, your respiratory system will have largely caught up, and you'll be taking in more oxygen, so you can switch from anaerobic down to aerobic, right? That's the idea. Short-term anaerobic coverage while your respiratory system meets your energy demand, so you can switch to your aerobic respiration, and that's your gold standard, right? That's when you hit your sweet spot. You also have another one where you have an enzyme called creatine kinase. And in this particular case, you have a phosphate on this particular molecule that you can actually transfer to ADP to create ATP. This is also low output, and it's a short-term deal. So you'll have enough of this to basically be able to kind of create a short-term kind of a stream of ATP, at least long enough to hopefully get you to the point where you're starting to get a little bit more better oxygen saturation, so that you can support your higher activity. And then there's other types of anaerobic respiration. For instance, where you go straight from glycolysis to fermentation. So when you're actually going from ATP to lactic acid, or excuse me, from glucose to lactic acid, and you generate ATP in the process, this is fermentation. You'll generate a little bit of ATP, not too much, right? Because from that first process, so when you're going from glucose to lactic acid, you're only generating maybe about 2 ATP per glucose molecule. By the way, if you're going through aerobic respiration, you're producing something on the order of 38 ATP per glucose molecule. So significantly higher efficiency if you're breathing oxygen, okay, which is the reason why you want to breathe oxygen. So the combination of these three, neither of which requires oxygen, working together in concert will allow you short-term coverage, at least enough ATP to cover you so you can run fast, quick, on demand, until you get your respiratory system and your oxygen saturation levels up to the point where you can kick into aerobic respiration. By the way, the field of burn compound, which I hate, is this is the lactic acid buildup. Remember we talked about that if you're in your kind of warm-up stage and you're working out and you feel the burn in your muscles and it's temporary, but it goes away, that's actually lactic acid buildup, which basically means you are burning anaerobically, which from a physiological perspective is absolutely useless. Because remember, if you're working out and you're looking for that really good, strong physiological benefit, which is what you should be looking for, that's why we love cardio and stuff like that, right? Is you're trying to figure out how to get into the aerobic respirations mode as quickly as possible. If you're basically feeling the burn before you get up to your oxygen levels, that's lactic acid buildup. That basically means you are inefficiently burning. It's anaerobic. Yeah. Because what'll happen is if you're exceeding your oxygen, then you're going to start to kick back into one of these anaerobic states. And so if you're feeling the burn, then you're overextending. You want to drop back because your oxygen levels are not supporting your levels. So you want to basically dial back your activity so that you don't feel that lactic acid buildup. If you have enough oxygenation in there, that should go away because it's temporary. But that's what you want. You want your activity level to match your oxygen saturation levels. That's your sweet spot. So here's kind of what it looks like. So this is what ATP production looks like in skeletal muscles. So in terms of the adenylate cyclase, in this particular case, you can see the adenylate cyclase, kinase, that's a different enzyme that I just mentioned. Forget about that one. Right, so adenylate kinase, I keep saying cyclase. So basically what's going to happen here is you have a phosphate on this guy who's going to grab onto an ADP, and it's going to basically smash a phosphate onto the ATP. And then it's going to create an ATP and in the process create an AMP. So this is the idea, right? This is the one you want right here. But again, River, it's just like one molecule at a time. So this is not high output, this is low output. In terms of the creatine kinase, you have a phosphate on your actual creatine phosphate. That's this piece of it. So what's going to happen is it's going to basically take this phosphate, put it onto this ADP to turn it to ATP, and then what's going to leave is creatine. Okay. And then typically what will happen is that'll go get phosphorylated again, and so you can recharge it, right? Anaerobic pathway, basically you're going to go through glycolysis. So this is your actual 9-step metabolic pathway that'll turn glucose into pyruvate plus 2 ATP in that process. But then you convert the pyruvate, because now pyruvate is a waste molecule. So you convert pyruvate into lactate, which is lactic acid. And then this is sent to the bloodstream, which then builds up in the muscles, and you feel it as that burning sensation. Okay. Which then, of course, as it circulates out, the burn will go away. But you have to stop replacing that lactic acid. So if you're on fermentation, then you'll feel that burn. That's the reason why, like, for instance, if I start off a little bit and, I'm kind of, sometimes I'm like, I get a little bit, adrenaline. I'm kind of like, right, I'm ready for this, right? I'm excited. Let's go. Right. Sometimes it's like, okay, now I'm feeling a little bit of burn. So what I'm going to do is I'm going to dial myself back a little bit, allow myself to slow down so I can kind of stop the anaerobic demand, stop the lactic acid buildup, and kind of give my respiratory system a chance to catch up. So I kind of back away from that a little bit. And then once I kind of start to feel good, like my breathing is there, like I've hit my point, then I kind of start to slowly, progressively kind of dial it up a little bit. I'll kind of dial it up, equilibrate, dial it up, equilibrate, dial it up, equilibrate, and then step it back down, basically. And of course, aerobic, basically you're going through the entire pathway, glycolysis, and through the mitochondria in order to create tons and tons of ATP. So that's your gold standard. That's the one you want. So what's muscle fatigue? So obviously what a muscle does is work, right? So basically when you have a decreased ability to do work, then that means your muscles are fatiguing. And so in this particular case, you kind of have imbalances in ATP, right? So for instance, you have depletion of ATP, obviously will cause muscle fatigue because your muscles just simply won't be able to contract, and you'll feel that as fatigue. Essentially, by the way, fatigue is a very general term. There's a lot of things that can cause fatigue. This is more of like a muscle mechanics version of it, but you could also have neural stimulus as part of a fatigue as well. So that can also be a thing. But you can see a decreased ATP production is causing muscle fatigue as well. Oxidative stress is another one. So when you go through metabolism like this, you generate byproducts of oxygen. So one of the things that people don't realize is that as you use oxygen, it actually spits off some really, really nasty oxidative byproducts called reactive oxygen species or oxidative stress types of molecules. And so what these guys will do is these come across as like free radicals and things like that, superoxides. So these are all really, really nasty oxidizing byproducts. Peroxide is another example. And so what does that mean? What it basically means is most of these guys will have an unpaired electron associated with them. So they're desperate to get an electron. And what that means is they'll rove around like a band of marauders, and they'll basically steal electrons from whomever they wish. The problem is if they steal those electrons from your tissue, they'll do damage to your tissue. If they do it to your DNA, they'll mutate your DNA. So these are really strongly oxidizing things. Matter of fact, we would be dead if we didn't have a strategy to defang these strong oxidizers. And so in our cells, we have specific organelles, peroxisomes, that are full of enzymes whose job it is to defang these oxidative byproducts. If we didn't have those peroxisomes in our cells, then basically oxygen to us would be about as lethal as cyanide is. And so that's the only reason why we can use oxygen, because we've got these peroxisomes that sort of defangs all of these reactive byproducts. So, so again, for instance, physiological contracture, which is a type of fatigue. Again, notice common theme here, right? Where you basically get a lack or a decrease in ATP. And so if you have a lack of ATP, you can't contract anymore because you need ATP to contract, right? It hydrolyzes into ADP, which you need, phosphate, which you need, all comes from ATP. You need ATP to actually relax because you need to detach. So if you have a lack of ATP, you have neither, right? So you don't have the fundamental hydrolysis cycle of ATP in order to do the power stroke and contraction piece of it, but you also don't have the ATP in order to let those cross bridges go so you can relax. So you get neither of them. That's physiological contraction when you're sort of stuck at that particular point. And so what you're waiting for is the next round of ATP production. So that's essentially what kind of gets you past this sort of fatigue. So that's basically contracture, where you're sort of frozen. Right? So that's basically contracture is, for lack of a better, more technical term, right? It's kind of like a physiological or anatomical freezing of something, some particular type of motion or activity. Psychological fatigue, it's all in your head, right? The Marines are really good at beating this one out of you, right? So you're running, you feel like you're about ready to die, and you drop to the ground. What do? The drill sergeant kicks you and says, "Get up and keep running," right? So they beat this one out of you. Why? What are they doing? Well, yeah, a little abusive, but what they're doing is they're beating the psychological fatigue out of you. They're saying, like, listen, you think you're at your end, but you're not, right? Physically, you can't be at your end. Why? Because part of the reason what basic is about, at least in those sorts of activities, is not to be abusive to the soldiers, but their job is to prepare the soldiers for battle, right? The idea is if you're on a battlefield and you're having to run and move, guess what? You don't have time when there's bullets in the air to get tired and to drop to the ground, because that could be the last thing you do, right? So it doesn't matter how tired you are, or how what that stomach or what that side ache feels like in your side. If you're on a battlefield, it doesn't matter. You have to run anyway. And so the thing is, rather than your body giving up on you in the battlefield, we'd rather give up on you on the training ground, so that we can condition your body to basically do more than what you think it's capable of doing, so that you don't die on the battlefield. Go ahead. Yep.
Which is the most horrific things that could possibly go through in a war, so that you can overcome and adapt to the situation wherever you come from.
The idea being, if I show you worst case scenario when I'm friendly to you, then you'll be ready for worst case scenario when it's not friendly, right? So you're right, it's a survival thing. They don't want you to come back in a body bag. They want you to survive. They want you to live. They want you to wear the medals at the end of the battle, right? They want you to come home and be decorated. That's what they want, right? That's the reason. That it doesn't seem like that when you're going through it, but that's the deal, right? So rigor mortis, we already talked about this one. Yes. So we already got that one. We already talked a little bit about soreness as well. So basically, right, with rigor mortis, essentially, you need that ATP in order to detach, right? So you're not making ATP anymore, so it's going to take you some time to degrade those cross bridges. But also think about it, your tissues are degrading across the board. So what does that mean? Well, another thing that means is the sarcoplasmic reticulum is degrading, right? So it's not holding the calcium quite as well. So all those things were designed to keep the calcium in the sarcoplasmic reticulum are degrading, so it's leaking out, which means you're getting a constant contraction signal, which favors cross bridge formation. So not only do you have the inability to detach the cross bridges, but you still have that persistent calcium signal in there that's favoring cross bridge attachment. So all that has to degrade, right? That's the reason why rigor mortis takes time to sort of break down, is because all that has to degrade and let go, and that takes a little bit of time there. So soreness basically is injury, right, at a micro scale, and of course the inflammation that comes along with it. Okay, that's basically what your soreness is. And the idea is you kind of build yourself back bigger and better, right? I mean, assuming you didn't damage yourself, right? Then it's kind of pointless, right? Because if you damage yourself, then you're not able to work out because you're waiting to heal, and which means that you're going backwards. So it's like, okay, now I just put myself on the injury list. So now I got to actually work out again to get back to where I started before I injured myself, right? So that's the reason why injury is always something that you want to keep your eyes on whenever you're training or doing anything like that. So basically, when you take a look at the idea of oxygen deficit, or oxygen debt, sometimes is what it's referred to. This is the phenomenon, that lag time that we're talking about, right? So between when you begin your exercise and when your oxygen meets your exercise, right? So this is your lag between the start of exercise to when your oxygen saturation is meeting the demand, right? So what you're doing is you're borrowing against the debt, right? Because you're saying, listen, do you think you could advance me a little bit of energy, because I got to be able to run and I don't have the oxygen levels to do that with? So it's like, sure, I'll go ahead and advance you. That's why they call it oxygen debt or oxygen deficit. And then once you start breathing and your oxygen saturation meets your demand, then on the post-exercise side of things, you have excess oxygen consumption, right? So this is basically the lag time on the back end. When you stop exercising, then there's a lag time before your breathing returns to normal, right? So if you've ever like run, like a mile, and or I'm not talking about sprinting, right? Just kind of like run a mile. So you want to get into that. It takes a little while before your breath gets in there, and that first lap might be like, right? And then eventually it gets a little easier, and you're like, okay, I can do this for a little bit, right? And then afterwards, you stop suddenly, and then you're what? Right? That is the post-exercise calibration. So that's that lag time. And then eventually what happens is that starts to slowly come down. Your breathing starts to slow down, broaden, and it comes back down to physiological. So your respiratory system is not an immediate, right? So what's happening here? So it's kind of like you borrow against the oxygen in the beginning, and then you're paying it back at the end, right? So that's kind of the way you look at it. So what is this doing then? So if you're not exercising, then why are you still taking in that extra oxygen? Well, because you have to basically restore everything. Remember, you are consuming your materials without oxygen. So we're paying you in advance. We gave you a little bit of an advance there, but guess what? You're going to pay that back, right? So when you stop and you still have a high oxygen saturation, even though now your physical activity does not demand the high oxygen, what are you doing? what you're doing is you're paying it back. You're restoring all that stuff that you got in advance for, and you're paying it all back to restore homeostasis. So that's kind of like this idea of the payback. Okay, so that's all skeletal muscle. Now we're going to talk very quickly about smooth muscle and keep moving. Cardiac muscle is going to be even shorter, actually, very short, right? Because that's cardiovascular, and that'll be AMP2. So smooth muscle is interesting, right? Because first of all, they don't have striations, which basically means no sarcomeres. Do they have muscle fibers? Yes, they do, but they're smaller. Are they shaped the same? No, they're more like gummy worms, right? Instead of long cylinders. Are they polynucleate? The answer is no, they have single nuclei. So they're kind of a little on the normal side, right? Do they have actin and myosin? The answer is yes, they do. It's not organized quite the same way, but they do. And actually they have more. Okay, so wait a minute. So those are some things in terms of comparatives to skeletal, right? But what are some things that are different than skeletal? Well, first of all, they have what's called caveoli. These are basically kind of indentations or puckers in the sarcolemma. And so these guys got to act as sort of like T-tubules. So functionally, they're related to T-tubules. They also have something else that skeletal muscle doesn't have, something called dense bodies. So instead of Z discs, which they don't have, they have dense bodies, and the actin is attached to the dense bodies. Now, unlike sarcomeres, which were largely just actin and myosin, that was it, right? There's also another type of filament, an intermediate filament, that is not actually involved in contractility. So there's an extra piece of protein structure. By the way, if you were working on you may not have caught this, or if you had general bio in the past, if you caught this on the review, intermediate filaments like actin and like microtubules are part of the organelle grouping that is called the cytoskeleton of every cell, right? Typically, intermediate filaments are structural proteins. They act like a scaffold in the cell that basically kind of hold the cell up. So they're literally like kind of a scaffold that kind of hold the membranes up, basically. Kind of like, I always like to think of it as sort of like the frame of a tent, right? The cell membrane is like the tent fabric. The intermediate filaments are like the frame of the tent. They basically hold the tent's basic shape, okay? Intermediate filaments are largely structural. So what we're saying here is that smooth muscle have this sort of structural scaffold protein that sort of holds its shape and acts as sort of like a structural backdrop. So what about calcium? Is that still required? Yes, it is, actually. In this case, what happens is there's another calcium binding protein that it binds to called calmodulin. And calmodulin will regulate another myosin regulating enzyme called myosin kinase. Now, this whole talk about kinase is a little confusing, right? Because, well, what exactly is a kinase? Kinases are simple. Kinases are basically a big family of enzymes that have one function. They phosphorylate things. They basically hang a phosphate on other things. Now, what's the effect of that? Typically speaking, when you put a phosphate on a protein, it has the effect of either turning it on or turning it off. So whenever you see a kinase, what it's fundamentally doing is basically taking a phosphate and adding it to another molecule. So oftentimes that has like a light switch effect on that other molecule. Or in the case of like our ATP regeneration, it was simply just to restore or rebuild ATP in that case. So ultimately, this is associated with the cross bridge formation that's part of the smooth muscle. Now, we're not going to get deep into the mechanism of smooth muscle shortening like we did with skeletal muscle. But basically, a kinase is a result of it. Now, how do you relax? Well, the relaxation is caused by the counterpart to a kinase, which is called a phosphatase. A phosphatase, basically, its job is to remove a phosphate. So kinases add a phosphate, phosphatases remove it. Makes sense. If the myosin kinase adds a phosphate, which is then what stimulates the contraction piece of it, then how do you remove that? With a phosphatase that then undoes it, and then you're able to relax. Okay. So this is kind of what it looks like. So there's our smooth muscle. That's just recapitulation. So it looks like Rocky Horror Picture Show, right? So basically you've got the fishnet stockings going on here. A little bit. Okay, so I couldn't help it. That's a, you know, we saw the SpongeBob movie, the original one. And when I saw this, it reminds, I don't know if you remember, if you saw that, first of all. But if you remember at the end when SpongeBob was doing his little guitar wizard scene, if you remember what Patrick looked like, he had the fishnet stockings on and the pumps. That was Patrick. Look, it's pink. It's got the fishnet stockings. It's like dead on. It's Patrick in the SpongeBob movie. So that's, sorry, I saw Patrick. Soon as I saw him, like, Patrick, there you are. Anyway, so you probably remember that scene now, right? It's hilarious because, I mean, he totally looked like something out of Rocky Horror Picture Show. But anyway, so basically this is what it looks like. So here you can see you've got basically these dense bodies, right? These little kind of intersections where you have your intermediate filaments. Some of these intermediate filaments are non-contractile, like for instance, you know, this guy here. So these are non-contractile. So they're basically kind of holding things together. And then when you take a look at the actual actin-myosin complex, it's these kind of like little green purple things. So you have these little actin pieces here, which still have kind of like a little bit of overlap with your myosin pieces here. So you still have the basic idea of an overlap system, right? So when they start to walk, that is when the myosin start to grab and pull, you're going to get sliding in this direction like that. So the sliding filament model is still kind of the same. It's just that you've organized it a little bit differently. So now instead of being between two Z lines, you've got it basically tethered like a hammock in between two dense bodies. You still have a region of overlap, and so you still have sliding. And so when all these start to shorten together, what's going to happen is they're basically just going to pull their little local area together. And so you're going to get basically what looks like this when you contract. It's going to be like a cinching. And it's not a lot, but remember, smooth muscle you're just looking at very small localized contractions. You're not looking at like skeletal muscle where you're looking at massive movements across space. Skeletal muscle, I mean, smooth muscle is just basically small local contractions at a very, very local level. And so something like this across many different smooth muscles is enough in order to generate the kind of shortening that you need. Cardiac muscle, on the other hand, right? This is only in heart. So it is striated. So that basically means it does have sarcomeres, but it's also branched, right? Which basically means that these guys are kind of interlaced between each other. Okay, so I'm not really an artist here, but we'll see that in just a second. Right, so they're kind of interlaced between each other. They're usually uninucleate, right? So that whole polynucleate thing was a weirdness associated with skeletal muscle. The important thing is, however, that they are bounded by intercalated discs. And intercalated discs are groups of gap junctions. What's a gap junction? I think we talked about this in the histology. So gap junction basically is like a sieve-like region of channels that connects two cells. So for instance, if I have two cells, cardiac cells, they're going to be connected by these gap junctions. So anything that happens to cell number one will immediately be communicated to cell number two. So you do this for coordinated contraction, right? Because heart muscles, unlike skeletal muscles, all contract in unison, right? So when you get the contraction signal, everybody, all the cardiomyocytes in your heart, from top to bottom, all contract in unison as soon as I get that. So that's different than skeletal muscles, right? So the only way you can do that is if everybody hears that contraction signal at the same time. The only way you can do that is if everybody's connected to the same cytoplasm. So they get that same signal at the same time. There are some significant difference between your action potential dynamics, which you typically look at in the physiology section of the cardiovascular section, but this basically produces longer refractory periods. Remember, these are the insensitive periods. The periods where they don't respond to another stimulus because they're busy resetting, right? They're repolarizing. So you've already sprung the mousetrap, so you can't respring it, right? Because it's already sprung. You have to basically reset it. And so until you reset it, it's insensitive. That's like the refractory period. So for cardiac muscle cells, there is a longer refractory period, which means it takes them longer to reset. So the important thing is you both need sodium and calcium influx for depolarization for cardiac muscle cells. And calcium, there's a slight difference with calcium. You don't just have endogenous calcium from the sarcoplasmic reticulum, you also have exogenous calcium, which comes from outside the cardiac cells. And so you have two different sources of calcium in cardiac cells. And like I said, the details of that, don't get too bogged down in it. Don't lose too much sleep over it, because that's a set of physiology in cardiac cells that you tend to unpack a little much more detail once you get to the cardiovascular section, okay, of physiology in the book. So this kind of should look familiar. So here you can see you've got your striated cells with your intercalated discs here. And then you can see how they are branched. So here's two cells with like a little fork right there. So those like branched cells. And this is kind of what it looks, so for the most part, looks more similar to a skeletal muscle cell than not. Right? Which makes sense, because I mean, if you already define the system and it works well, you don't really need to reinvent it. Right? Ages. Gotta love it, right?
No.
Yeah, you're like, how about no? So the effects of aging, we've already talked about a lot of this, haven't we? Right? We already talked about, I mean, you get reduced muscle mass as you get older, right? Just kind of a thing that happens, right? Just everything gets less efficient. That's pretty much the story of everything. Your contraction response gets a little bit sloppier, right? So that's kind of like, a lot of times in older, like your response times are slowing down, right? So there's that's happening at a physiological level, not just at kind of like a perception level. You can't last as long, right? So you can't, you don't have as much duration or stamina as you used to. So your muscles fatigue faster. And so they kind of break down a little bit. It takes longer to recover. So if you injure your muscle, that's actually true for all wound healing, actually it takes a lot longer as you get older. So a lot of these aging effects isn't really specific to muscles. It's just, there's a muscle version of this, but it's pretty much ubiquitous across all tissues itself. You'll lose muscle fibers, right? Especially the fast twitch. Remember, the fast twitch ones are those sprinter muscles, right? Because the idea is like, when you're 80, you're not sprinting, right? If anything, you're walking, right? You're walking. Yeah. Okay. So that's not a fair comparison. I mean, probably most people don't run as quite like Carl Lewis, but yeah. But instead of running the 5K, you're walking the 5K, right? So loss of muscle fibers, right? That's the fast twitch stuff. And then of course your vasculature, that's muscle supply. I mean, the blood supply to the muscles is also atrophying. So basically aging, if you look at it, it's not like a specific thing. What you're looking at is like just a global general sort of a decline in the efficiency of a lot of processes and things like that across the board, right? This is one of the reasons why we don't find like, oh, this the magic, the aging gene. We can change the aging gene, then we're going to live forever. It's like, no, we're not, because aging isn't just aging or a thing. It's just basically a universal, just kind of a tailing off and a decrease in efficiency over time globally of all the systems in your body, not just one system. They'd have to find that probably that covers everything in your body, which would be hard, hard to do. But so here's kind of like our little, in this case, it's muscular dystrophy, right? So you can see how the different systems are tied together, right? So how you basically have nervous components, lymph and immune components, cardiovascular, respiratory, urinary, digestive, and skeletal, how they all kind of feed into what we typically think of as largely just a primarily muscular type of a situation. And the only reason why I show these is just to kind of give you a little bit of a snapshot, not to dig into in depth, right? Because that starts to move into pathophysiology, and that's not the role that we have right now. But it's just to kind of give you a little bit of an overview of just like, when you think about a particular disorder, almost never is it as simple as like, oh, well, this is just affecting your muscles. No, right? Physiology and your systems are so integrated that usually there's symptoms in a lot of systems in your body. Okay, so that integrated idea is a thing. Okay. Finally, the end of chapter 9. The beginning of chapter 11. Okay, so nervous system. So there's going to be a lot of things in the nervous system. We already talked about. So when I hit those slides, expect me to move fast, right? Because I don't want to basically spend time redoing a lecture that we've already done. I'm just going to add a couple of new pieces to what we've already have. Okay, so that means you want to hang on to things like neuromuscular junction, action potential, all that sort of stuff. Okay. Okay, so let's go ahead and take a look at the nervous system itself. Now, the nervous system, basically, when you take a look at the nervous system, is basically an integration system. You got all this stuff in your body. Now you got to figure out how to integrate it. And that's what the nervous system does. Actually, you don't have just one integration system. You have two of them, right? Because we end up with nervous system in AMP1. In AMP2, we start off with another integration system, the endocrine system, which is a chemical or hormonal integration system, which tells you a little something, right? Whenever it's not redundant, it's important. Right? Whenever you have two integration systems, that's basically screaming at you, this is a big deal. Control, regulation, integration, right? That's the name of the game. And so it just means that those ideas, those topics, control and regulation, are super, super, super important, that you're spending double the money on two of them, okay? So let's go ahead and take a look then at our nervous system. Let's kind of break it down a little bit. We got a little bit of this from the tissues chapter, right? So basically the tissue types that we see in nervous tissue are going to be pinned down by, first of all, neurons. And the second piece of the neural tissue is glial cells, right? Or support cells. Now, the neurons or the nerve has some specific pieces to it. So this is kind of a little bit of a glossary exercise here. There's some terms here, some things here that I want to define and unpack so you kind of know what they are when we're talking about them, right? So when we take a look at the concept of a nerve, generally speaking, we're looking at a bundle of axons that's either outside the brain or the spinal cord, right? So that's basically what a nerve is. So you're outside the brain or the spinal cord. Now, generally speaking, that when you take a look at nerves, there's really two global families of nerves. The first one are the cranial nerves, right? You have 12 of them. By the way, these are the ones that we are already studying in lab. Remember, so some of you saw the olfactory bulb, some of you saw the optic nerve in your sheep brains. Those are 2 of the 12 cranial nerves. And so we'll be learning all 12 of them in lab. But the reason why they're called cranial nerves is because they originate from the brain, but they're outside of the brain, right? Like the optic nerve, for instance, is originating from the brain, but it's actually going into the eyeball, right? So that's basically how you perceive the images. The other one is spinal nerves. So these are basically nerves that originate from the spinal cord. You have 31 pairs of these, right? So when you guys looked at the spinal cord model, you'll notice that you have basically the roots. You have the dorsal root and the ventral root. How do you know the dorsal root? Well, the dorsal root has that little swelling on it, right, called the dorsal root ganglion. And then they fuse together to create your spinal root. And that goes on to become the spinal nerve. And there's 31 pairs of those that kind of go up and down your spinal cord, and they kind of radiate out through the rest of the body. Yeah, so the spine, well, no, not the sheep brain. So the spinal nerves are coming off the spine. I'm thinking of the little plastic spinal cord model. Yeah. So a ganglion, what you'll see is basically a collection of neuron cell bodies. Typically, it's outside the brain and the spinal cord. So these are like processing centers. A plexus is a network, in this case, of axons. It kind of looks like a fishnet. By the way, the term plexus is generic. This is a neural plexus, right? So which means you have a network of axons. You can have a vascular plexus where you have a network, a fishnet-like network of blood vessels, right? So a plexus is just simply a fishnet-like network of something, okay? In this case, we're looking at a plexus. Typically, it's outside the brain and the spine, which is part of the central nervous system. And then, of course, you have glial cells, which are your support cells. So they basically support the neuron. So there's a couple of terms there, and we'll kind of basically unpack a couple of more. So what does the nervous system do? Well, obviously, its job is to maintain homeostasis. So basically regulate, this is the big one, and coordinate your physiology. Those two are prime words. Like, for instance, if I were on an MCAT and they were asking me what is the purpose of the nervous system, I would definitely be looking for words like regulate and coordinate physiology. That's what it does for a living, right? That's what these integration systems are doing. They're regulating and coordinating physiology. So one thing that it'll do, the way it does this is it'll always receive sensory input. So basically taking in information from your outside world. Then it'll be integrating that information as it comes in. And obviously those integrating centers are going to be your brain and your spinal cord, right? That's your central nervous system. And then typically there will be some sort of a response associated with that integrated information, usually involving controlling muscles or glands or some sort of a response. Okay. Of course, mental activity, what you're doing now, consciousness, which Luke, you were discussing about how it was amazing that we're even aware or conscious. Consciousness is a thing. It's an amazing thing, actually, if you think about it. Basically, every night we die a mini death, literally, right? We essentially go unconscious and then we come back, right? I mean, that's something we do every day. So it's kind of an amazing thing. Thinking, memory, emotion for us, right? So this is something that your nervous system is all in control of. Okay, so now let's take a look at the major divisions. And this is going to be kind of helpful in terms of organizing. Let's sit back here next to you. So it's kind of helpful to organize things, right? So first of all, you have two big watershed sides of your nervous system. Your central nervous system, which is your brain and your spinal cord, is largely what we like. That's kind of what we're studying in lab right now, right? So we're kind of studying mostly the central nervous system stuff. But then outside of the brain and spinal cord is what's called your peripheral nervous system. And your peripheral nervous system is everything outside. So all of those spinal nerves, the stuff that's out in your periphery, your sensory receptors. Remember all the sensory receptors you talked about in the skin? Remember we talked about the deep pressure, we talked about the light pressure, the tactile corpuscles, the heat and cold sensors. So all of those are all part of the peripheral. Okay. So now basically when you take a look at this, so we're going to break this into the components for the divisions in the branches. I want to focus on some of these guys because it's kind of help us sort of organize some things. So when you take a look at the central nervous system, it largely is what it is, right? Your brain and your spinal cord, it's the integrator. So this basically integrates stuff. This is command control. So this is your processing center. Okay. Now, the peripheral nervous system can be broken down into several divisions depending on what you're trying to get accomplished. Generally speaking, in the peripheral system, you're looking at things like receptors, nerves. Remember, that's the bundle of axons. Ganglia, remember, that's the bundle of cell bodies. So those are like little micro processing centers that are kind of out there in the periphery. And plexities, remember, those are those little neural networks that are kind of spread out throughout your body. Generally speaking, you can break your peripheral into two different divisions, either your sensory division or your motor division. So your sensory division basically is kind of what it says it is, right? So this is the part of your peripheral nervous system whose job it is to collect information from your outside world. So you have all of these sensory receptors out there, and it's collecting information from your outside world, and it's bringing all that back into where? Your central nervous system, your brain in particular, so that your brain can integrate it, make sense out of it, sort through it, and figure out, okay, what does this all mean exactly? So the direction of that is going from the outside in. So we refer to that as afferent, or I like to think of it as arrival. That is to say, arrival to where? To the brain, right? So the afferent direction is basically going to be arriving to the brain. Having brought in information from the outside world, it is now arriving to the brain through the afferent system to the brain to basically give it all the information it collected. Now, what's going to happen after that? The brain is going to sort through all of this. It's going to figure out what to do with it all. It's going to make some sort of a decision, and then it's going to send that out in terms of some sort of response. So then what it does is it goes through the motor pathway. This is mostly what we've been studying in the muscle. The neuromuscular junction is the motor side of the peripheral nervous system. So this will then exit through the efferent pathway. Efferent for exit, E and E. So it's exiting with the decision that the brain has come to. Like, for instance, if it's a muscle, that decision is to contract, right? And so then it will basically go through and deliver that signal to the muscle. Now, from the direction, we come to the branches of the peripheral nervous system. And so from the efferent side of things, we're going to leave the sensory side alone for a little bit here. So from the efferent side, because it's kind of where we were in NMJ in the last chapter, you have two major branches of the peripheral nervous system of the motor side of things. The first one is somatic, which, if you think about it, means body. Soma for body. So basically, the somatic is going to be a voluntary controlled situation. This is essentially going to be your skeletal muscle. So when we are basically sending a contraction signal through the motor division of the peripheral nervous system, through the efferent pathways that are exiting the central nervous system, heading toward the skeletal muscle, it is going to be following through the somatic branch, which is voluntary. You can choose or not to contract your muscle, and it will be targeting your skeletal muscle. So the neuromuscular junction is going to be found down here, the interface between this neuron of the motor neuron and the skeletal muscle. So this stimulates what? Contraction. That's what we learned in the last chapter. However, the efferent system also has what's called an autonomic branch to it as well. This is involuntary. And what this one is controlling is largely your visceral reactions. That is to say, all of those organ activities. So you break that into two different groups, two different subdivisions, either sympathetic or parasympathetic. And one of the theories what I want to do in the central nervous system is to compare and contrast sympathetic and parasympathetic. So that is a discussion that I want to have come hell or high water before the end of the semester, is so that you have a clear conception of what sympathetic and parasympathetic is doing, because that'll set you up, because this is what's innervating the visceral reaction. So when you're talking about the movements of your stomach as you're digesting, or the movement of your heart, or the breathing of your lungs, this is all being controlled by this part, this branch of your efferent pathway. Okay. And it breaks down into either sympathetic or parasympathetic control. Well, sympathetic control generally has a nickname to it. Its job is largely to upregulate cardiac and smooth muscle and, of course, glands, and it readies you for physical activity. So we tend to call this one the fight or flight. This is your stress response, sympathetic is. So if I let the lion in the room, you're going to drop into sympathetic mode because you're getting ready to fight or flight. Unless, of course, you have moose. Then moose will take care of it, right? Right. So, or yeah, we'll just let William take care of it. So the rest of us can just relax. We'll be like, don't worry, William's got it. Yeah, William's got it. So the other one, parasympathetic, is going to downregulate your cardiac smooth and glands. And so basically it is resting. So this is what's called your rest and digest mode. So really, when you look at it fundamentally from a visceral, what you honestly see from visceral control regulation is you basically see a toggling between two different physiological states, sympathetic and parasympathetic. As a matter of fact, as you go through the nervous control of each of your visceral organ systems, as you go through AMP2, oftentimes that discussion of nervous control is a comparative between the sympathetic side of it and the parasympathetic side of it. Okay, so here's your nervous system, lots of nerves. There it is. Okay, that's good. So this is one of those quick slides, right? So here's kind of like your motor division versus your sensory division. So basically you have your descending motor division and you have your ascending sensory division, right? Because it's coming from the periphery of the body going up to the central nervous system, whereas the motor division is descending. It's going from the central nervous system out to the rest of the body, to whatever effectors are out there, right? And so this is just kind of like more of a flow chart and a visual way of depicting exactly what the previous table was depicting. So this is essentially a recapitulation. It's just organizing it more in a flow chart sort of a way. Right? The only reason why I put the slide in here is because some of you guys like the flow chart look to things. Some of you guys prefer the table. Okay, so that way at least you have one of the two. So one of these two hopefully will kind of click and make sense. Okay, so let's take a look at the sensory side of things first of all, before we start moving into the motor side of things and kind of dig into some of the broader components of the nervous system and some of the physiology of the nervous system. So remember we said the sensory is going to be the afferent, right? That's the arrival. So this is going to be transmitting your action potential from your receptors to your brain. And so basically this is collecting everything out there in your skin. For instance, if you're kind of feeling something, you feel smoothness or pressure or heat or whatever it is, that's all coming external stimuli that's basically coming through your sensors up to your brain or your spinal cord. And then that's the efferent pathway. So this is the ascending efferent pathway. And then of course you have your motor afferent pathway, efferent pathway, which is your exiting or descending pathway, which is going to be basically taking the communication that your central nervous system came up with, and it's going to be taking that message out to the target effectors, right? So that's what the effectors are. These are the target cells that need to do some sort of action. In this case, in the case of the muscles, that action is to contract, right? So your somatic, which is largely what we talked about in the last chapter, by the way, you didn't know that, but we were largely talking about the somatic nervous system in taking essentially a voluntary signal, an action potential, and voluntarily sending that through a motor neuron to the neuromuscular junction where we get the contraction of the skeletal muscle. So that entire discussion that we had, that entire flow from action potential to muscle contraction is essentially the somatic nervous system. Right? That's essentially where the somatic nervous system. Now, autonomic where we said is going to be sympathetic versus parasympathetic. So it depends on whether or not you're going to be at rest or having physical activity. So essentially what's going to happen with the sympathetic is you're going to be preparing your body for physical activity, which basically means you're going to be upregulating oxygen. You're also upregulate ATP production and everything else that you need for physical activity, right? So that basically requires an upregulation of everything. So increasing your ATP production, you're going to increase your mitochondrial activity, which means you're going to have to increase your oxygen consumption in order to maintain muscle contraction, which is your increased activity, which is going to require a lot more ATP, right? So basically, you're getting ready to run or fight in the case of but either way, you're going to need more ATP in order to overpower the lion, and that's your sympathetic mode. But at the moment, you guys are relatively at rest, so you don't need any of that. So your oxygen levels basically are calibrated for you at rest. And so essentially you're calibrating and just regulating what you're doing right now. Digesting, for instance, what you're doing. If you ate something before class, that's what you're doing right now. If you have to urinate and emptying your urinary bladder, basic physiological functions, day-to-day activities, no stress, nothing like that. These are the activities that are happening in the parasympathetic mode. Now, there is an off branch of parasympathetic, which I'm going to call 2A, which is called enteric. And so the enteric nervous system is actually a subdivision of the parasympathetic. So as it turns out, there is a lot of control and coordination with your digestion. There's a lot of hormones involved in your digestion. There's a lot of enzyme production in the pancreas associated with digestion. There's a lot of coordination that has to happen because your GI tract is one long tube, but every part of the tube is doing something different. Your esophagus is doing something different than your stomach, which is doing something different than your intestines, which is doing something different than your large intestines. And so everything is doing something very different. In order to coordinate all those activities to get good, smooth transfer and flow, you have to have a lot of control and regulation. So you have a lot of hormones produced in the digestive system. You've got a lot of nervous impulses that is coordinating the muscle contractions of your digestive system. Your stomach alone is like a little butter churner. I mean, it's just basically churning away. So those muscle contractions are controlled separately by this enteric nervous system. As a matter of fact, what happens is you've got a cranial nerve, the vagus nerve, number 10, that's part of the parasympathetic group. And you'll learn about the vagus nerve. But basically the vagus one is like the grand poobah of all 12, because the vagus nerve is in control of all the visceral organs and their activity. So the vagus nerve is the one that's kind of governing all those visceral organs that you learn about in AMP2. So it's a really, really big deal. But when it gets to the digestive system, what the vagus does is instead of controlling all the minutia of all the control and regulation of the digestive system, because there's a lot going on there, what the vagus nerve does is it basically just kind of like it outsources it. It kind of subdivides it out to the enteric nervous system, kind of creating almost like a semi-autonomous nervous system in its own right. And so the vagus system just basically says, listen, enteric nervous system with your little plexuses and things like that, why don't you just take complete ownership of the digestive thing? There's a lot going on there. I've got to deal with the lungs. I've got to deal with the heart. I've got to deal with the kidneys, the bladder. I've got to deal with all the rest of the organs. So why don't you just basically take some autonomy and you just basically take charge of the digestive system? And we call that sort of semi-autonomous de facto nervous system, the enteric nervous system. It's actually a subdivision of what the vagus nerve does. So the vagus nerve kind of like parts that out. And so you'll learn more about that when you get to the digestive system. Okay, but first of all, before we get to that, let's take a look at the actual tissues themselves, right? Let's learn a little something about the neurons. So the neurons, remember, this is kind of like of our 4 tissue types. This is the excitable tissue. Remember, you had the contractile tissue, which was muscle tissue. You had the connective tissue. This is the excitable one. So when you take a look at the neuron, it actually has a couple of major components to it. The first one is the cell body. We already talked about the cell body, right? We already said that a cluster of cell bodies is called what? A ganglia, right? So basically the cell body will essentially do most of the functions of a cell, right? These are all these typical cell functions, right? So for instance, protein synthesis, all the housekeeping stuff, There are structures inside the cell body called nissel bodies, which is essentially rough ER, right? And so you get this in the biology review. And what does rough ER do? It does protein synthesis. In the case of a neuron, that protein oftentimes could be, not always, but could be a neurotransmitter. So the cell body is doing exactly everything that a normal cell would do. The other part of a neuron is called the dendrites. So these are little extensions attached to the cell body. And so if you take a look at the cell body, it's usually like, this is a motor neuron, but it looks something like this. So this is your cell body. You have nissel bodies, which are these like little things that are like around the nucleus. And then the dendrites are these little projections. And what these guys are doing is they're receiving information from sensory receptors. So there's information coming in through the dendrites and actually taking it to the cell body. Remember, the cell body is the processing center of the neuron. So it's bringing these messages into the cell body where the cell body will process these incoming messages. It'll come up to a decision and it'll send that decision out the axon. So the axon is basically a long process that extends and heads toward its connection point or its target. Now, generally speaking, in this area right here, you have what's called the axon hillock. So this is an area of the cell body that kind of forms this like little cup-shaped structure before you get to the axon. So if this is your axon, then this is like your axon hillock. Think of it sort of like a drain or a funnel, right? The open area at the top is the axon hillock. And as it starts to kind of drain down into the axon, it starts to move toward its target. So that's like the initial zone. We call that the initial segment because this is where the action potential starts. This is why we call it the trigger zone, right? So the action potential is that initial opening of the sodium channels that then gets that depolarization, that rolling depolarization we talked about in the last chapter, where you just kind of start tipping over one voltage gated channel over another, over another, another. That first tip domino happens here. So this is your trigger zone. Tipping of the first domino. Now, inside the axon is going to be fluid called the axoplasm, right? This is like the cytoplasm of the axon. And then the axolemma is the plasma membrane of the axon. And then eventually this will lead down to the terminus of the axon where we see the presynaptic terminal full of synaptic vesicles, and those synaptic vesicles are full of neurotransmitter. At this particular point, this looks familiar, doesn't it? Because we saw this on the neuromuscular junction slide. That's where we were, right? So this kind of connects it to the other side of the neuron. So this is kind of what it looks like. And so what I'll do is I'll start off with this image, and we'll take a look at some transport mechanisms as we sort of start to flesh some of this stuff out, okay? Which I might just start with the picture, since it's always good to start off with the picture. That sound good?
Yes, sir.
So I think we are a little beyond our witching hour.